Hi, my name is Kelsey Dahlgren, and I am a biochemistry PhD student at the University of Colorado Boulder in Jeff Cameron's lab. In the Cameron lab, we study cyanobacteria, which are a large group of related bacteria that, just like plants, perform oxygenic photosynthesis and carbon fixation. And just a quick review on that, oxygenic photosynthesis is when the cell absorbs light energy from the sun and produces oxygen from water, and carbon fixation is when the cell takes in carbon dioxide from the air or water and uses it to make sugars. And cyanobacteria are actually really important ecologically and industrially. Ecologically, cyanobacteria are very common in the ocean and other aquatic environments, and they perform about 30% of the global carbon fixation every year, and they produce about 25% of the oxygen we breathe every year. In industry, they're very important because you don't have to give them sugars to grow on. You just need to give them air. So say you wanted to make the same chemical, but you could try and make it in yeast, or you could try and make it in cyanobacteria. The yeast, you would have to feed them to make them grow. However, cyanobacteria, you basically just give them some air and they would grow. So they're a little bit cheaper and more sustainable to grow and produce chemicals of value. And my project in the Cameron lab on cyanobacteria is understanding photosynthesis and carbon fixation, but through the lens of spatial organization within the cell. And the reason why I want to sort of get a better understanding of photosynthesis and carbon fixation is because just generally understanding the physiology of cyanobacteria could definitely improve, for example, metabolic engineering. Understanding how the cell works would give us a better understanding of what sort of pathways we need to avoid or use when we're trying to metabolically engineer cyanobacteria to produce biofuels. It could also just be used to increase something as simple as increasing the growth rate of cyanobacteria. And far out in the future, if we wanted to, for example, make a photosynthetic cell from scratch, we would need to understand all of the reactions that go into photosynthesis and carbon fixation and how they are spatially organized within the cell. And just to give you an idea of the spatial organization of cyanobacteria, it's not like you would expect for bacteria, because you know that bacteria do not have any membrane-bound organelles, if you remember that from high school biology. So you would think that they basically are just a big homogeneous mixture of proteins and DNA held in a cell membrane. Cyanobacteria are not like that at all. They have a lot of organization. So they have the outer membrane on the very outside, and then a cell wall on the inside, and then an inner membrane inside of the cell wall. And all of those protect the cell from the outside world. And just like plants, cyanobacteria actually have thylakoid membranes. So plants have thylakoid membranes in their chloroplasts. Cyanobacteria just have thylakoid membranes. And these basically run along the edges of so that there's the inner membrane, and they run along the inside edges of that, along the outside of the cell. And the cell usually has quite a few of them. And then there's also the carboxysome, which is used for carbon fixation. And you can kind of think of the carboxysome like an orange. There's a protein shell, which is like the peel of the orange, and the fruit of the orange is the enzyme that performs carbon fixation. And the cell uses these carboxysomes to concentrate carbon dioxide, since the enzyme that performs carbon fixation is sensitive to oxygen. So the cell artificially increases carbon dioxide there, so the enzyme will use carbon dioxide instead of oxygen. That's being produced by the photosynthesis happening in the thylakoid membranes. There's also another very interesting part of the cell, and it's basically, if you think of the cells as like a rectangular prism with some rounded edges, the thylakoid membranes go along the edge and meet in the corner. And in this corner, it looks like the thylakoid membranes are being made when the cell grows and divides. It also looks like the photosystems are assembled in this spot. So that's a very long process, and photosystems are the proteins that the cell uses for photosynthesis. And it also appears that the carboxysomes, the orange, the orange-like protein structures, are also produced in, or are assembled in the corner of the cell. So there's a lot of stuff happening in this cell corner. And what I want to understand is why the cell would spatially put all of these reactions in the same spot. Is there a reason that the cell wants to assemble the carboxysome and build the membrane and assemble the photosystems in the same spot? Or did it just happen by coincidence? And the way that I want to go about answering this question is to understand all the proteins that are functioning in this cell corner. And I hope to do that through proteomics, which is basically just the study of all of the proteins in the cell or in a specific area of the cell. And I plan to use proximity-based labeling proteomics in the cell. And basically what that means is that I am going to target a small protein that I put into the cell called Apex to this corner, which I can just do by attaching it to a protein that already localizes to the cell corner. And my enzyme Apex basically will take in a substrate that I put in and catalyze a reaction to make that substrate very, very, very reactive. So when the cell, or when the enzyme releases the reactive molecule, it basically wants to stick on to anything nearby. Labeling all, or most, hopefully, of the proteins nearby with this molecule. And this reactive molecule actually can act as a handle for me to pull these proteins out from all of the rest of the proteins in the cell that don't have the handle. And basically, because I'm able to pull them all out, I can then identify them using mass spectroscopy, which is just a method that uses the mass of proteins or molecules to identify them. And once I've identified the proteins, hopefully in the corner of the cell, hopefully I can do some analysis to figure out what proteins are important for the structure of the corner of the cell, and what reactions are actually being carried out in the corner of the cell. And hopefully I can use this knowledge to better understand cyanobacteria physiology, especially the spatial organization of the cells, and hopefully be able to improve growth rates or metabolic engineering of cells in the future. Does anyone have any questions?